blinding up here. Okay, so what I'd like to do is talk about it's a very generic title, Analytical Methods for Investigation of the Neurovascular Space. So I want to talk about some of the work that we're doing with the blood-brain barrier, and then some of the work that we're working on with the nitric oxide, Proxy Nitrate detection in single cells, and then lead on into how we're trying to do this in a awake, freely roaming animal. But first, I wanted to congratulate Christy. I took this picture back in 2009 at the Gordon Conference on Microfluidics, and it looks like she's started to really do a lot of microfluidics work. So congratulations, and a very well-deserved award. So the Neurovascular Unit has a large number of different kinds of cells, and it's actually, it seems like the theme today is sort of inflammation and neuroinflammation. So these cells are all involved in the neurovascular unit: endothelial cells, neurons, astrocytes, and microglia. And today I'll talk mostly about endothelial cells and then macrophages as a sort of precursor to looking at eventually microglia. And some of the analytes we're interested in is looking at peptides, neurotransmitters, both amino acid neurotransmitters and catecholamines, nitric oxide, and then reactive nitrogen and oxygen species. So the blood-brain barrier, it used to be, 5 years ago, you just show the endothelial cells with the tight junction, and you would say that was the blood-brain barrier. But really, the blood-brain barrier is fed by this neurovascular unit. So the astrocytes provide nutrients, sort of like the quick shop for the endothelial cells, and then you have the neurons in the space that are affected by things that are transported or by release of nutrients and things from the astrocytes. And then also there's the immune cells in the brain, the microglia on the brain side, and then there will also be macrophages on the blood side of this neurovascular unit. So what I'm going to talk about today is a little bit about a mimic that we use for looking at transport of compounds across the blood-brain barrier, bovine brain microvessel endothelial cells, and then I'll continue with what Chris was talking about, looking at single cell analysis for reactive oxygen and nitrogen species. So the endothelial cells that make up the blood-brain barrier regulate the transport of substances across the blood-brain barrier. So in contrast to peripheral capillaries, these cells have very tight junctions that only allow a small number of molecules across the barrier, and almost everything's actively transported or has to be really lipophilic to get across that barrier. It also has a metabolic barrier, and I'll show that in a minute, where peptides and other compounds that come to the blood-brain barrier can actually be converted into peptide fragments that might have completely different physiological functions than the original peptide. So it acts as both the physical barrier and the metabolic barrier. And also, because it has endothelial nitric oxide synthase, it produces nitric oxide, but it can also, as I'll show in a little bit, it can induce nitric oxide synthase at this barrier as well through cytokines and other inflammatory agents. And so we mostly work, because we're in Kansas and we have lots of cow brains in Kansas, we've been using the bovine brain microvessel endothelial cell in vitro cell culture system model. Basically, the students go to the nearest slaughterhouse, pick up some brains, and then they isolate the capillaries and the bovine brain that make up the blood-brain barrier, and then you can culture those out in a cell culture system and get a primary culture for the blood-brain barrier. So one example I wanted to show is we've done a lot of work with substance P, and more recently we've been working with Dynorphin. And we've been looking at this for two reasons. One, it's an interesting peptide in terms of pain and neuropathic pain, but also we have some collaborators in medicinal chemistry that are looking at modified versions of Dynorphin to look at both drug addiction and neuropathic pain. And so one of Courtney Kuhnlein that was in my group did this work on this Dynorphin A1 through 17, and she's now working with Ryan Bailey. And one of the things we were interested in is for the native peptide, when it reaches the blood-brain barrier, how is it metabolized, and what fragments might get across the blood-brain barrier and send a signal from the peripheral tissue into the brain. And so it's a 17 amino acid peptide. It has a messenger sequence, which if you do anything with opioids will look very familiar, and then a receptor preference on the other end. So we've done these Dynorphin metabolites two different ways. This shows initially she was complexing the peptides with copper, we had the hope that we would be able to form nice electroactive complexes and be able to do this by either microchip or capillary electrophoresis. It forms nice complexes with copper, but they're not very electroactive, and they tend to really stick to PDMS, so we've sort of given up on that. But she was able to get all the different dynorphin peptides separated using the copper, which actually helped resolve a couple of the peptides. And so this shows the separation just using capillary electrophoresis with UV detection. So then Courtney used this method to look at the metabolism of these peptides in a variety of samples. She took some blood. This shows the rat brain slices, and she also looked at spinal cord. And she has a lot of different data, but I really just wanted to focus right now on dynorphin 1 through 6, because she saw that in both the spinal cord and the brain slice. And so we were sort of interested in what's the transport properties of this 1 through 6 peptide at the blood-brain barrier. So here shows the production of the dynorphin 1 through 6 by the blood-brain barrier endothelial cells, so you can track over time the metabolism of dynorphin 1 through 17 into 1 through 6. You can also track the other peptides as well. Now this work was actually done with LCMS. So to do the transport study, you can take these cells that you've cultured from the cow brains and the endothelial cells that have been isolated from the cow brains, and then culture them on polycarbonate membranes, and then you actually get a blood and a brain side. They will grow so that they polarize, so you can look at studies or you look at transport across the blood-brain barrier. So you basically use a side-by-side diffusion cell. You can put the peptides on one side, and then they'll be transported across to the other side. You want to have a temperature-controlled system, and I'll show that in a minute, but if you go to 4 degrees versus 37 degrees, you can see whether it's sort of an active transport process or a passive transport process. And so you can use this setup to actually look at the transport of drugs and peptides and other substances across the blood-brain barrier. So when she did this, it looks like dynorphin A1 through 6 will pass either direction quite well. So it can go apical to basolateral or basolateral to apical. The permeation coefficients are about the same. But one of the things is that there is a temperature dependence. So if you go down to 4 degrees and basically shut off all the energy from the system, you don't see you see that the permeability is very much decreased. This indicates that it's either a receptor-mediated or endocytotic process, probably because it requires energy. And we haven't looked at whether there's a dynorphin. Well, actually, this would actually even bind the opioid receptor. We've shown that substance P actually is receptor-mediated, and we found the receptor for substance P in the blood-brain barrier microvessel endothelial cells using a Western blot. We haven't done that yet with the dynorphin, but that would be the next step, is to see if it's receptor-mediated or endocytosed. So here's some of the different, this just shows the different mechanisms of getting things across the blood-brain barrier. Very few things actually get across this tight junction. Lipophilic compounds will go through this transcellular lipophilic pathway. And there are certain proteins that will, there's transporter proteins that will bring amino acids. For example, if you eat a lot of Chinese food and you have a lot of glutamate, you don't want it all to rush into your brain. You'd like to have a transport protein that would regulate that. But the dynorphin is either by this receptor-mediated transcytosis or adsorptive transcytosis. One really interesting thing, and part of the reason why I wanted to talk about this 1 through 6, is not only does 1 through 6 go in both directions quite well and gets transported into the brain and also from the brain into the peripheral tissue, but another thing that we found is that it actually affects the permeability of fluorescein. So we use fluorescein as a non-radioactive marker of blood-brain barrier permeability, and normally it should have very low permeability across the blood-brain barrier. But when you add dynorphin 1 through 6 into the media, you actually see an increased permeability of the fluorescein across the blood-brain barrier. So you can see in both directions the permeability is increased. And then if you go to 4 degrees, to look at whether it's receptor-mediated or not, you can see the permeability is decreased. So this is a really interesting result. And Audis's group had also seen this with Lou and Keflin, so it's sort of interesting. There must be some sort of receptor. There must be some mechanism where these compounds bind to the blood-brain barrier and not only get transported into the blood-brain barrier, but also somehow affect the tight junctions and allow more compounds to get in. And Jonathan, if you have any good ideas why this is, I'd be glad to hear them. So that's just an example of sort of the endothelial cells. And when we've been working on those for quite a while. And one of the things that is of interest is how nitric oxide can sort of open up the blood-brain barrier. And I'll show in a minute some really old data from Audis's group that shows that nitric oxide does increase the permeability of blood-brain barrier, and that cytokines can induce nitric oxide synthase in those cells. And then I'll talk about some of the work that we're doing to look at reactive oxygen species generated from nitric oxide and superoxide, either in endothelial cells or microglia or macrophages. So this is an old thesis, and I apologize that I was going to try and redo the table. But the first thing that Vicki Thiel did in Ken Audis's group was just use some nitric oxide donors and just look to see if it increases the permeability of the blood-brain barrier. And you can see the control has a permeability coefficient of about 5.21, and when you add the nitric oxide donor, it goes up quite a bit. So it is opening the blood-brain barrier, and this is artificially doing this with a compound that generates nitric oxide. So to tie into Christie's talk, if you take a cytokine and incubate the same blood-brain barrier, or the BBMEC cells for 24 hours, you can see that you are increasing the amount of nitric oxide that's being produced. So you're inducing this nitric oxide synthase in the blood-brain barrier cells. And then lastly, if you add this the cytokine and compare it to the control and just look at the permeability, I believe this is mannitol or sucrose, you can see an increase in permeability due to the induction of what we believe is the induction of nitric oxide synthase at the blood-brain barrier and causing this increase in permeability of the barrier. So this led us into this whole nitric oxide thing, because back here, and this was several years ago, they were doing the nitrite levels with the grease assay, and we thought, well, maybe we can do something in chip format that could make things more high throughput and more accurate. So we started looking at nitric oxide, nitrite. Actually, we started with nitrite and nitrate, and now we're looking at nitric oxide and proxy nitrate. But in all these studies, we're looking at the direct electrochemical detection of these compounds. And hopefully Professor Amatori is still in here, but he's done quite a bit of work at looking at these directly with electrochemical detection. So there's two ways that we can do the electrochemical detection for the capillary or microchip electrophoresis. Most of what I'll show right now is using an isolated potentiostat and sticking the electrode as close as possible to the end of the channel, basically sort of in the channel. So we call it in-channel detection, but it's right in the very end of the channel. And that gives us the best separation and efficiencies with the lowest sort of overpotential that we have to apply to the electrode. And it allows us to, because it's an isolated potentiostat, we can still do the separation without killing the potentiostat when we apply the potential for the separation. So one of the things that we did is we wanted, for Kasi de Silva, who is a visiting professor in my group, and Dulan, wanted to look at the detection, first of just nitric oxide by microchip electrophoresis electrochemical detection. Now it's neutral, so you might wonder why we want to just detect a neutral compound with microchip electrophoresis with electrochemical detection, but we want to be able to separate it from nitrite and proxy nitrate, and we want to know where it comes out. And so he just took some proline nanoate and reacted it, and you can see you get a nice peak for NO in the electrophoregram, and you can actually follow. This is the proline nanoate, this is nitrite, and then you get the nitric oxide production as well. I think this might be off. I think this is nitrite. So the other thing that we're really interested in, and this is partly not just because of Dr. Audis, but Ken Audis, but there's also another colleague of ours, Christian Schoenig, that's very interested in peroxy nitrite and oxidative modifications of proteins and looking at nitrotyrosine. And so Celeste Frankenfeld in my group was working on this nitrate detection system, and she got really interested in the Proxy Nitrate. Can we detect Proxy Nitrate? And so one of the things that she found that was really interesting is that when you buy Proxy Nitrate from Cayman Chemicals, it comes in a very high pH buffer, and it's stable. So it's in a solution in a high pH buffer. So one of the problems with chromatography is you could never do a separation on a normal column with a buffer at pH 11 or higher. So, but if you do capillary electrophoresis, you can actually stabilize this Proxy Nitrate and separate it quite well from nitrate and nitrite. This is just using UV detection. Now we could separate these and we could do the analysis, but the detection limits, because it was UV detection, were not very good. So then we started looking at, can we detect the Proxy Nitrate electrochemically? And so this shows an example of the same Cayman Chemicals Proxy Nitrate, which we just put in the buffer reservoir and injected over and over again. And it will degrade over time. You can see it's actually quite a bit of time that's here. But it will degrade over time, mainly because there's a lot of carbon dioxide in the buffer reservoir, and that will cause this reaction to occur over time. But you can see, we can definitely detect the Proxy Nitrate here. And then we could use a SIN1, which is a Proxy Nitrate generating agent. We just looked at that to see if we could monitor Proxy Nitrate generation over time. So we took some of the SIN1, stuck it in the reservoir, and then you can continuously monitor the Proxy Nitrate. And then this will eventually turn into nitrate and nitrite. And so this shows just the plot time of Proxy Nitrite from SIN1 over time. And the main thing I wanted to show here is with the microchip, we can do a lot of injection like every minute on this device, so we can monitor the production fairly quickly. We did have to change the buffer reservoir, so that's what these steps are, is when it's just changing the run buffer in the buffer reservoirs. So that leads me to what Chris was talking about. So we thought, well, if we can stabilize Proxy Nitrate at a high pH, so all those separations were done at a high pH buffer using T-tab and reverse polarity, then maybe we could actually detect Proxy Nitrite in single cells. And so we got really interested in this, and fortunately Chris is just down the road, and he's an excellent scientist and an expert on single cell analysis, so we teamed up. And the idea here is to be able to detect both NO and Proxy Nitrite in a single cell using his device. And if we can, the idea is if we can lyse the cells very quickly at a high pH, the intracellular Proxy Nitrite would be stabilized. And since the separation is very fast, less than a minute, usually we should be able to stabilize it long enough to actually detect the intracellular concentration of Proxy Nitrite directly, also NO directly. So we shouldn't have problems that you have with the interferences with the fluorescent dyes. So we have done some experiments with macrophages. As Chris indicated, they can be activated by, they can produce both nitric oxide and Proxy Nitrite in their adherent cells. This will look familiar because I think it's the same one he used, but we've been doing bulk cell lysate. So basically we incubate the macrophages with LPS for 6 to 8 hours, then harvest the cells. He does the harvest very quickly, lyses them very quickly, centrifuges them, and injects them directly into the chip. So one of the first things he had to do was see what else is electroactive in these cells. So this shows just a cell lysate. This is one of the activated cells. So you can see, we see nitrite in the cells. We actually get a peak for tyrosine and glutathione, and we can also detect the nitric oxide in the cells. Now, because we're taking these bulk cells and they're just the cells are just going in the sample reservoir, the NO is continuously going into the air and degrading. So basically, what you see is a decrease in the nitric oxide. But when we do the single cell analysis, the idea would be to lyse here and then immediately get an NO concentration of the amount of nitric oxide in that single cell. So the second thing that we've done is, and this is very preliminary, but we took LPS and interferon gamma and then added PMA. So what this does is this produces a nitric oxide. This produces the superoxide, so we should get Proxy Nitrate in the macrophages. And then what he did is he harvests the cells, you lyse them in CE run buffer, which is about pH 11 buffer with T-tab in it, so it's going to lyse the cells very quickly. He centrifuges it, and then this is what we see. We can see actually a peak that we think is Proxy Nitrate in this stimulated lysate. Now, we've only done this a couple times, so we're still trying to figure this out. But one of the things we did, Dulon did do, was do a voltammetric characterization of some of these peaks. And this peak, it has very similar electrochemical properties to Proxy Nitrate. One of the future goals would be in the single cell analysis system is to actually have a dual electrode system, so we could do on-the-fly voltammetric characterization, so that we'd have not only a migration time, but also some information about how easy or difficult it is to oxidize, just like scanning voltammetry, so we could say with greater certainty that that's indeed what it is. So we're working with Chris, and the next step on our end is he's going to help us integrate the electrochemical detector into a single cell analysis system. And so then the idea would be to lyse these cells that are in a really high pH buffer and try and measure nitric oxide and Proxy Nitrate and separate that from ascorbate and glutathione and tyrosine. Maybe look for some other, we can also, actually at that pH, hydrogen peroxide actually separates from nitric oxide because it is slightly ionized. We're at such a high pH. So you can actually look at several reactive oxygen species and nitrogen species simultaneously. So so we talked about endothelial cells and macrophages. We want to move from macrophages actually to microglia once we figure out how to do the single cell analysis with the electrochemical detection. Then we would really like to be able to look at single microglia in terms of how they're activated, and there's different phenotypes based on how old an animal is or what part of the brain the microglia might be isolated from. So we'd really like to be able to look at the effect of phenotype on the production of nitric oxide and Proxy Nitrate in microglia. So the last thing is just another thing that we're working on, which is sort of, so we have the endothelial cells, we have the microglia, macrophages. So what about the astrocytes and the neurons? Well, if you put a whole animal together, you get the intact neurovascular unit. And so we do a lot of microdialysis sampling, and that allows you to continuously monitor what's going on in the brain in near real time. And I just wanted to show a couple of examples related to blood-brain barrier permeability and in the neurovascular unit. So one of the experiments that we did a while ago was to use this online microdialysis, microchip electrophoresis with online derivatization to look at simultaneous, the goal of this is to look at the release of amino acid neurotransmitters and blood-brain barrier permeability at the same time. So we've done some preliminary experiments. The students graduated, and we're now working up to get another student that they're at a point where they can do the animal studies. But the idea here is, as I mentioned before, fluorescein is a good blood-brain barrier permeability marker. And actually, Bill Emquist from Minnesota has used it a lot to look at changes in transporters in blood-brain barrier permeability. So what we did is we gave the rat an IV infusion of fluorescein, and so you can watch it reach steady state in the brain, the amount of fluorescein reach steady state in the brain. And then we can use the on-chip derivatization with NDA cyanide and fluorescence detection to monitor aspartate and glutamate and other amino acid neurotransmitters. Now, this is done with an anesthetized animal, so there could be some changes in the blood-brain barrier with that as well. One of the things you can see is this just shows the extracellular fluid of the brain of the rat obtained from the microdialysate. And you can see after this infusion, you see the fluorescein, and you can also see the aspartate and glutamate. And so this is just a rat, anesthetized rat. We haven't actually done any ischemia yet, which is what we really like to do. Because when we do ischemia, we'd like to see if there's changes in this, if this increases, and what's the timing between the time the aspartate and glutamate increase and then the opening of the blood-brain barrier. Is there can we figure out what the sequence is and the timing of that is? And if we want to verify that this peak is indeed fluorescein, we can just switch the laser or the wavelength that we're monitoring So this is fluorescein, and then we have the other amino acids. The other thing we've done is actually wanted to do on-animal sampling, so look at, and some people call this Frankenrat, but what we wanted to do is look at, let the animal run around and be able to do on-animal sampling. And so we have, in the drug delivery area, they use osmotic pumps a lot to deliver drugs to the brain. So we just use this osmotic pump as a pump for the microdialysis system, and then just have it a collection vial on the end of the rat. So the rat could move around. It's got this little sort of weird helmet thing on, but the pump is actually driven by osmotic pressure, so there's no battery, no you don't have to plug it in. So it's just all self-contained system. And we use this to look at Ldopa transport across the blood-brain barrier. So we gave the rat a dose of Ldopa, and then you can look at some of the metabolites showing up in the brain: DOPAC, HVA, and 5-HIA. And so this looks like, oops, see if it's, oh, something got lost here. Anyway, oh, there, oh. So we're trying to move from that rat to on-animal analysis, and I'm going to talk about this Wednesday morning. But this is our lab on a sheep project, and the idea here is to develop a integrated microfluidic system with the microdialysis pump, the high voltage power supply, telemetry, and a potentiostat to continuously monitor neurotransmitters in awake, freely roaming sheep. Now we picked sheep because we can't make this small enough for rats yet, and sheep are pretty docile. And they've actually, as you'd probably suspect, in New Zealand, they've done a lot of work with sheep and behavior. So we could take advantage of some of that literature. But the idea is to put this thing on the sheep and be able to continuously monitor neurotransmitters. Right now, what we've been working on, and I'll talk a little bit about this on Wednesday, is just doing transdermal microdialysis on the sheep, because we don't want to do brain surgery until we're sure the whole system works. So we've been putting a microdialysis probe in the sheep, putting the system on the sheep, and then giving it a histamine injection and trying to look for the formation of nitrite from the histamine nitric oxide response. And this actually shows just some peaks of nitric oxide and peroxide obtained with this system. Okay, so in conclusions, I hope I showed a little bit about some different studies that you can do concerning the neurovascular unit. You know, the BB-MEC transport studies are a very useful screening method for looking at metabolism and transport of peptides and also other drugs. We've been looking at the electrochemistry of nitric oxide and proxy nitrate, and we've been looking at this in bulk cell. We're working with Chris to try and do it in single cell. And then we'd like to do the in vivo monitoring of blood-brain barrier transport. So in the future directions, we want to hook up our electrochemical detector to Chris's single cell analysis. We want to look at some of the peptides like Dynorphin and substance P along with cytokines on the effect of nitric oxide synthase at the blood-brain barrier. And also some of the fragments that are being generated at the barrier itself. We'd like to use the fluorescein amino acid combination detection system to look at ischemia and nitric oxide release and amino acid neurotransmitters. And then lastly, we've been working on animal sensors for correlating neurochemistry and behavior. And I just put this here because sort of our ultimate goal is to really look at the microglia, which they were here somewhere. I can't. Oh, here they are. So, you know, to look at these microglia, which are activated by a lot more things than macrophages, because if there's head trauma, if some albumin gets leaked across the blood-brain barrier into the brain, it will activate the microglia. There's a lot of different things that will activate microglia to produce nitric oxide. And there's also a belief that when people are in the process of aging, the microglia are sort of hyperactivate or it's on a constant alert as people get older, and so that might be a source of oxidative stress in aging and Alzheimer's disease. So I'd like to thank the people that did the work. Courtney's here, right here. She's working with Ryan Bailey now at the University of Illinois. She did all the Dynorphin work. Dulon and Giuseppe were working on the nitric oxide in single cells, and Matt Hulvey did a lot of work with Proxy Nitrate. Annie and Dave and Ryan were working on the on-animal sensors. And then Emily Mines is now working for Nancy Albritton, and she was also working on the nitric oxide work with Chris and myself. And then I'd like to also thank Chris's group and Fracassi da Silva, who is on a sabbatical in my group, who did a lot of the electrochemistry of the nitric oxide compounds. People help with microdialysis, and then Pinnacle Technologies helping us build the on-animal device. And then also thank NIH and American Heart Association for their support. And I was going to give this to Jonathan. Oh well. And so you should go to the Adams Award on Wednesday, which Jonathan Sweetler's getting, and this is a quote that Buzz Adams had in his lab, posted in his lab when he was at KU, and I just think it's a good quote, so I sometimes like to end my talks with it. So I'd be happy to answer any questions.
Okay, we are starting a recess.
Oh,
and
right.
If anybody have questions, I think we have time to answer a few.
It's like the hot lights here, so. Yeah, Dana.
When you do those permeability studies with that little blood-brain barrier, the two chambers there,
Yeah.
does that allow access to, say like they have your endothelial cells grown on some sort of membrane?
Yeah.
Do you have access to be able to go in with like a couple small electrodes and actually?
They do measure the tear. Yeah, they do measure. But I don't know if they actually, we haven't done that. I know Ken's group measures the resistance. She usually just measures the integrity of using fluorescein or mannitol or sucrose. But I know they measure the tear as well. But I think they may have a different device with the membrane. So it's grown on a membrane. So I think they put it in some other device to measure the resistance. But yeah, that's a good point. That's another way to measure how tight those junctions are.